Good morning, everyone. Thank you so much for joining the NEC Society's webinar on modeling NEC. We are working very hard to keep patients and families centered in all the work that we do here at the NEC Society. So we're really grateful and honored to dedicate today's webinar to Charlie. And Charlie survived NEC after she was diagnosed when she was 5 weeks old. She was born prematurely, diagnosed with NEC, and lost 70% of her small intestine. Today, Charlie is 3.5 years old, and her family describes her as silly and creative and playful. And she loves to dance and play soccer and go to preschool. And her family is obviously very proud of her. So we are grateful and honored to dedicate today's webinar to Charlie, and just really want to recognize all the families that make the NEC Society possible, and the clinicians and the researchers who care for babies and families like Charlie. And again, they make our work possible. So thank you so much for being here, and we're going to get started. So for anyone who's just getting to know the NEC Society, welcome. We're so happy that you are here. The NEC Society is a 501c3 nonprofit organization that is dedicated to building a world without NEC. We bring together patient families who've been personally affected by this disease, along with researchers, clinicians, and other people who care. And we work hard to advance NEC research, education, and advocacy so that we can prevent this devastating disease and improve outcomes for all infants and families. We recently developed a prioritized research agenda, which basically means we brought a lot of people in our community together to figure out what research gaps really are perhaps the most impactful and urgent questions that we really need to be focusing our limited amount of time and resources and funding towards. And so a lot of the work that we're going to be doing in the coming months and years are going to be focused on these research priorities. So you're going to be seeing them quite often in the work that we do. The NEC Society is so grateful to be a member of the CZI Rare as One network. We are one of 50 nonprofit patient-led organizations that make up the CZI Rare as One network. And it's really been a transformational opportunity for the NEC Society to be supported by Rare as One and to be part of this network. And we are very fortunate to have two of the CZI Rare as One team members here today they're going to tell you a little bit more about the Rare as One network and how lucky we are to be part of it. So here are your speakers for today. If I haven't had the pleasure of meeting you yet, welcome. I'm Jen. I'm the founder and director of the NEC Society. I lost my son Micah to complications of NEC just before his first birthday. Micah developed NEC when he was 6 weeks old, had multiple bowel resections, lots of complications, but did come home, and we thought he had overcome his most challenging obstacles, but then tragically developed pneumonia and passed away just before his first birthday. So I'm so grateful and honored to be able to do this work and to collaborate with the amazing people that we have on today's webinar, including Andra, Julianne, Hala, Misty, and Steve. So I'm very excited that you're going to hear from them in just a moment. And just a quick reminder before I hand it over to Andra and Julianne that the NEC Society never is providing medical advice on this webinar or ever. We don't provide medical advice, and all of our faculty views are independent from the NEC Society as an organization. So thank you so much for being here, and I'm going to hand it over to Andra now.
We have Science and Society, and that is where Rare as One lives. So I shared with you the mission of CZI across when we're talking about CZI science. We believe that supporting science and technology that will make it possible to cure, prevent, or manage all diseases by the end of the century. Yes, that is what we call an audacious goal. But we really do believe that with strong innovation and doing things differently and including people in the process in a way that's never been done before, that this goal is possible. And when we're talking about people, that means those of you who are actually affected by the diseases. So within this, we started Rare as One. Rare as One is a network of what is now, as I mentioned, 50 rare disease organizations. We believe that providing minimal funding and key capacity building resources, training, and expertise that organizations like NEC can and are moving mountains in a way that we haven't seen before. We really recognize that to do things, organizations need capacity. I mean, organizations cannot get this work done alone. As you all know, you work together as a community, and it's nonstop. There's so much to learn. There's so much to think about. So many support resources that are needed in your community, and all of that takes capacity. So that's really where we started this first of its kind capacity building program, so that we could really identify the gaps in the rare disease space and provide groups with those trainings in these gap areas. We created, and we'll talk a little bit more about it, some fundamentals: finance, operations, hiring, fundraising, strategic planning. There's a program that really helps the organization build capacity around those areas. And you'll hear from Julianne a little bit more about a science advising program that we're still working on, and also a mentorship program to help leaders like Jen really take the knowledge they have and accelerate it. You may recognize one of the individuals in the top of the right-hand corner here is Lindsay with NEC, and we're so grateful to bring together the NEC team as well as the rest of the network this June for the very first time, based on COVID. We had our in-person convening. It was really fantastic. And what we were able to see, what we knew, that patients are really driving the research. Organizations like yours are uniquely poised to really get at building a diverse and inclusive patient community. You all are the gatekeepers to the trust. And as a community, you can also galvanize scientific collaboration, working with your patients, working with the physicians. We really believe that collaboration is what advances science in a way that has not been done before. You all are the best ones to articulate the disease insights, as Jen was just mentioning to you. Finding what was the most important to your community and really focusing the research on that is truly key to making meaningful advancements in a much faster way that has been done before. And I'm sure you all know that requires a lot of research dollars. And so NEC, like these other groups, are all spending a lot of their time and energy in major fundraising campaigns. So we believe, though, again, that if we provide minimal funding to help build these organizations, that you all then can really take that capacity and, as I had mentioned before, move mountains. And with that, I'd like to pass it on over to my colleague, Julianne Mills, who is also part of the Rares One team.
Thanks so much, Anda. Really appreciate it. And it's an honor to be here speaking with all of you today. So we've taken a sort of an incubator approach to the Rares One network, as Andrea had mentioned. I'm just going to go over into in a little more detail, sort of our technical approach to it. So what we've issued to our grant recipients is an award in the amount of $600,000 to be distributed over a period of 3 years. And there are specific goals that we've asked our grant recipients to aim for with those funds. We're asking them to build and expand their collaborative research network, to work with both their physician, clinician, and patient communities to create a shared research agenda, and to then host and lead a patient-led scientific convening across their community, and ultimately leading to and being a partner to the other grant recipients to help build the network. So far, this has been tremendously successful. As you heard Jen mention, and Andrea alluded to, our first convening was this past June, and just the dynamic that we saw happen across the organizations was truly proof to us that building a network virtually can transform what happens then when you have an opportunity to meet in person. And if we go to the next slide, please, Andrea. So there's a very specific way that we approach capacity building. The theory behind this is that if we provide a grant to do some research, if that research project doesn't work out, we haven't really helped to move the needle or drive any patient-led research. And instead, the way we approach the funding is to do some capacity building. That includes capacity building from an organizational perspective, like Andrew mentioned, through fundraising, board development, and sort of the really essential administrative needs for strong organizations, but then also helping to build out scientific capacity. How do you grow your research network? How do you reach consensus on what clinical guidelines or treatment guidelines might be? And then how do you host logistically? How do you host a patient-led scientific convening? And so we do all this through 3 goals: strengthening our communities, like I said, building capacity, and then promoting collaboration. We strengthen our communities by, providing that sort of landscape assessment and ongoing support. The building capacity is something that we do actually through all of our grant programs, not just those with the Rare as One, providing both that scientific capacity building as well as the organizational capacity building. And then promoting collaboration, we do that through things like we have an online chat forum, as well as those face-to-face meetings. And what we've learned is that some of our organizations sometimes don't have access to larger grants or larger funding opportunities because they are perceived to not have a strong organizational foundation. And so again, that's why we don't fund just one-off research projects. It's why we make an investment in these organizations to truly build themselves out as organizations so that they can continue to do the hard work and maintain their credibility as a patient-led research organization. If you go to the next slide, and the proof is in the pudding, if you will. From the first 2 years of this program, those first 30 grant recipients, we have some initial data. We'll obviously be doing this at the end of this year as well, so hope to revise this with some new numbers. But what we've seen is through those first 30 grant recipients, they have been able to successfully grow their research network to the tune of almost 600 researchers. They've had over 70 international scientific meetings, have been able to come together and create 26 consensus agendas for research, developed registries as well as biobanks, have gone on to win additional research grants, have developed industry partnerships, and even supported clinical trials. So what we found is that by providing that capacity building, both in terms of organization and science capacity, those modest improvements can have really transformational and exponential impacts. And if we go to the last slide, just to acknowledge that, we couldn't do this alone, not only without our rare disease partners, but Andrew and I are part of a larger team within CZI led by Tanya Simoncelli on the program team, and our partners on the tech side led by Sam Scavaner. So thank you again for your time today, and I'll pass it back to Jen and our next speaker.
Thank you both so much, Andrea and Julianne, for being here and for allowing the NEC Society to partner with you all. It's just such an honor that the NEC Society is part of Rare as One. Hala, you're up. We're excited to have you.
All right. Good morning, everyone. I would like to thank you all for joining. All right, so my name is Hala. I'm an associate professor at OU Children's. And before I start sharing with you about the science that's occurring in the lab, I would like to share with you why we're passionate about necrotizing enterocolitis. And it's some personal story that has occurred and has molded itself throughout the years. But I did my training back home in a tiny little country in the Middle East, and actually moved to the United States to continue pursuing research in newborn sepsis. But early during my training, I came back after a day being post-call, and one of the infants who actually was doing wonderful, former 25-weeker, doing really great, has passed. And these are the stories that a lot of us as trainees probably stay with you for a long time. And that there's an internal guilt that probably all of us have been experienced, which is, did I miss anything? Was there anything that I probably would have been able to detect? And one of the faculty, after we've talked, actually told me something which stuck with me, which is, this is NEC. It occurs in some babies. We can't do anything about it after it is struck, unfortunately, small intestine, and sometimes we can't do anything about it. And that has changed my research passion, actually, and my career toward looking into how can we prevent necrotizing enterocolitis throughout the years? How can we help the intestine of a preterm infant to develop faster after birth and prevent the predisposition to developing NEC? So to be able to do that, we had to go back and we had to think of what is the small intestine of a fetus at the third trimester? What does it usually do? And it's literally being exposed continuously to amniotic fluid, tons and tons of growth factors that help and promote active growth of these cells and dividing and actually, elongating the intestinal epithelium and changing, differentiating into, some cells that actually help the immune response and protect it against pathogens and bacteria. But unfortunately, and then we all know about the role of TLR4 from David Hackham's lab, where these intestinal epithelium in utero is coated with TLR4, which is very important for intestinal development in utero. Now, after birth, unfortunately, the preterm infants, the initial trigger, and I call them trigger because these are predisposing factors that make the intestinal epithelium of a preterm infant more at risk of developing NEC. The biggest thing is there's no amniotic fluid. The intestinal epithelium of a preterm infant is actually not exposed to the amniotic fluid that has all the growth factors and the stem cells and all the good stuff. And also we're in the NICU, antibiotics, feeding, all of those actually has this recurrent stressors that predisposes the gut to injury. But perhaps the most important factor is that now, instead of the intestinal epithelium of the fetus at the last trimester having, to grow in a homeostatic environment, now it has to protect itself from the biggest challenge, and that's the bacteria that actually coats the intestinal epithelium. This is a slide that shows the villi, which is the intestinal epithelium. In blue is the nucleus, and then the red that you can see literally touching the epithelium almost is the bacteria. And unfortunately, we all know about the pathological bacteria that actually can coat or actually can grow in the preterm intestinal lumen. So that has shaped, again, our interest in what we're doing right now, which is to identify something, strategies or some intervention that can help the preterm intestinal epithelium grow or mature faster postnatally after birth, and maybe shape the microbiome or the bacteria that's present in the lumen of the intestine into something more beneficial or healthy, instead of the pathogenic bacteria that we see that predisposes infants to NEC. So we turned into something that works, the human milk. We all know human milk has tons and tons of bioactive components, but one that has been kind of neglected a little bit is hyaluronan or hyaluronic acid. It literally is what we use on our faces and what people use in the joints, except that it's in a different molecular weight or size. And there's been quite a bit of literature initially from Cleveland Clinic about the importance of HA in human milk, especially early in lactation, in the first 2 weeks of life. And this is a period of life that if we need to do anything to prevent NEC would be crucial. This is the time that we can intervene to maybe help the intestinal epithelium have a little bit more defenses against bacteria and the injury that can occur. So HA has been used in multiple models of intestinal injury, but more like an adult intestinal injury. So with the help of Carol Delamont from Cleveland Clinic, we tested it in a very tiny pilot study. So this is the Stephen McElroy model, which you're going to hear about in a little bit, where it's a double challenge where we ablate Paneth cells, and which are very important for the innate intestinal immunity. And then we follow it up with a large bacteria load of Klebsiella that has been implicated in clinical necrotizing enterocolitis. And by giving HA35 3 days prior to this neck challenge, we were able to show that the animals had less gross intestinal injury, which is by eye, you see it less inflamed and swollen. And then you have histologically, there was less intestinal injury. And then the most important findings was that if we give HA35 just 3 days prior to the neck injury, the pups were protected from mortality. There was less percent mortality in the animals that received HA35. What we also found is that just giving HA35 was associated with improved permeability. So this intestinal epithelium was not leaky anymore from the neck injury, and bacteria wasn't able to cross from the lumen into the blood, which made these pups sick and die. We took this a little bit step backwards. So instead of just looking at HA35 in a neck injury model, the fact that it's present really high in the first 2 weeks of lactation in humans made us think, what does it do for postnatal intestinal development? So is it important for the growth of the intestinal epithelium after infants are born? So we gave HA35, also this is all in mice, for about 7 days. And after that, we looked at the intestinal development, just histological methods. So I'm not going to bore you with the details, but what we found was that just giving 7 days of HA35 without doing anything to the mice pups was associated with this accelerated or faster development of the intestine. The villi looked healthier, they were longer. The crypts which had all these dividing cells was deeper, and there was more Ki67, which is the proliferating cells that make up the intestinal epithelium. And then there were higher numbers of differentiated cells, goblet cells and Paneth cells, both of which are very important for protection of the intestinal epithelium against the pathological bacteria or bacteria in general. We also have some data about the microbiome that I didn't add because of time. Now, something that we're all asked all the time is that this is in mice. So what? How is that going to change practice? And more importantly, there's been quite a bit of talk about animal models and how they're not perfect. So we always have to go back and ask ourselves, does this happen? Would this happen in the intestinal epithelium of humans or primates specifically? And if we want to move research forward, obviously we need to do animal experiments. But again, we have to revert back. Would this happen in the human at the same time? Now, because of, because we need much more data before we actually test HA35 in preterm infants, we reverted to the simplest method at this point, which is testing HA35 in intestinal epithelium from preterm infants or preterm non-human primates. And this work was done by Kathy Burge, where she was able to isolate intestinal crypts from samples from preterm intestinal epithelium and then grow it into 3D organoids. And this, as you can see from the panel at the bottom, it literally is a ball or a spheroid that eventually will bud, and it has all the intestinal epithelial structure. Granted, there's no other cells and no bacteria. However, our question was pretty simple. What does HA35 do? And is it protective and facilitate the healing of the intestinal epithelium, which is the first line of cells that would be exposed to the injury? Now, the problem with this, in addition to the lack of other type of cells, is the 3D enteroids. Actually, the epithelium is inside. It's a ball and the lumen is inside. So to be able to access, to make it a little bit more physiologically relevant, this is just an example of how the inside is where you want the lumen. So when you're testing, you're not necessarily in a physiological manner. So to be able to access the epithelium, this is what we've done, is we've disrupted these 3D structures and grew them as monolayer on transwell so that we can expose whatever we need to, just like HA35 on top. And these are just examples of the 3D monolayers where you can see the structures, and the construct is to show that there's multiple types of cells, not just epithelium, but also in this case, it's the goblet cells are present in these 3D and this 2D monolayers from enteroids. So this is just an example of what we've looked at HA35 and the monolayers from preterm intestinal epithelium, and we've done a mechanical scratch. So it's an injury model, but it's a mechanical injury model with a plastic pipette tip. We just scratch the epithelium and then we test HA35 with different concentrations. And we've actually found that similar to the murine, so the mouse intestinal epithelium, HA35 actually facilitated healing, in this case, the migration of the cell to help healing of the wound faster than controls, which was very exciting because then we can move forward, hopefully, with the animal models when we've shown that it is also helpful in the human intestinal epithelium. Most recently, we've moved into some other type of injury. Obviously, the preterm intestinal epithelium is not exposed to mechanical injury, but actually to recurrent episodes, most likely of hypoxia, in addition to some bacterial products. So we've optimized a method where, if you remember that ball with the lumen inside, we've been able to flip it outwards so that we can expose whatever we need from the outside. And then we challenge it with hypoxia and either TNF or LPS. And hopefully we're going to move to heat and inactivated bacteria and live bacteria soon. And I just want to show you the bottom panels. This is the control where you have the lining of the villi in red, just supposed to be bright and healthy looking. And then you have these linked EK adhering that links the cells, and then the nucleus in blue. When we do the combination of hypoxia with TNF or LPS, you lose that wonderful red coating, and then the structure of the EK adherent or the linkage is disrupted, which is pretty similar to what you'd see in intestinal injury models or human intestinal epithelium. And then you can see also the cell viability, there's decrease in cell viability when we do the combination of hypoxia with the cytokines, whether LPS or TNF alpha. This is just a magnification. Again, there's disruption in how the villi would look like, and then disruption of the EK adherent. And there's more data from Kathy Hunter's lab that shows that there's increased permeability actually in an apical out neck, what we call neck in a dish, which I know there's limitations in them. With that, I would like to finish with acknowledgements with the OU Neonatology Lab, Kathy, Jeff, and Adam, who've been instrumental in all the animals in addition to the organoids. Obviously, recent collaborations with Misty and Stephen, and the funding. And that is it.
Awesome. Thank you so much, Hala. It was a wonderful presentation. Misty, we look forward to hearing from you.
Thank you so much. Just getting my screen up here. There we go. Let's see. Start from the beginning. Thank you for your patience. Can you see my slides okay?
Yep, perfect.
Okay, great. Thank you so much. I'm really grateful for the opportunity to present some of our work today that we're trying to move mountains, as Andrew said at the beginning, really looking at advancing the necrotizing enterocolitis models and really the importance of defining the disease. And I think as we learn more about NEC and we can build better models, and efforts in our lab are really focused on doing that as well. We're very grateful for all of our funding disclosures, which I have here, but in particular, a shout out to CCI for the NEC Society Rares 1 award, which has been really instrumental in getting our centers for the NEC biorepository up and running. So we're very grateful to you for that. And first, I wanted to start off the talk just telling you why I do research on NEC. As Hala also mentioned, we certainly have so many stories that of patients and families that have really touched us, that they were doing fine and then we come back in the next day and they're not there anymore and they passed away overnight. And I think as a neonatologist, unfortunately, we see NEC all of the time. And it's one of those diseases that really the entire clinical team is completely helpless. And I know the families and certainly the babies feel helpless as well, but the medical team also feels helpless. And we really have little to offer in terms of new treatment approaches over the last 30 to 40 years, because nothing's really changed. And so one of the big goals in our lab is really to help, how can we push this field forward in a transformative way in order to be able to provide some hope for our patients and their families. Because when we're faced with a diagnosis of NEC, we don't know how fast they're going to progress or how severe their situation is going to be. And so that's really one of the big focuses in our lab. So just a little brief outline of the talk, and this image is courtesy of Erin Umberger, who does amazing graphics. I'll talk a little bit about our bench and to bedside and back approach to NEC, how we're working to define NEC phenotypes using multi-omics approaches, and just introduce briefly our gut on a chip and microphysiologic system that has really built this NEC on a chip model, and how we're using that as a drug discovery approach. And really, this slide is what sums it up. So what we're really trying to do, and there's several preclinical models of NEC, and we don't have time to talk about them all. And Hala and Steve will do a great job talking about theirs as well. But really, this is how we model NEC in the lab. And so what we try to do is take what we know at the bedside from our babies and then bring that back into the lab, and then bring that back to the babies. That's the goal, is to really develop therapeutics for the disease, and ultimately preventative strategies to really build a world without NEC together with the NEC Society and all of you. And so when we're looking at these different preclinical models, we certainly use the mouse model of NEC, which I'll talk a little bit about, but then also, how can we reinvent that mouse model? Or, there's various other models, including the rat and piglet models as well, and certainly in vitro models, as Hala and I will both talk about. Well, she already talked about, but I'll talk a little bit about today. And one of those things is really the importance of biobanking. And something that we started in 2017, which has been transformative across several centers, is really biobanking the samples and really deeply characterizing and analyzing those samples from a multi-omics approach, so that we can learn more about the disease, but then also inform all of the animal models that we're all using in the lab to really get us better answers and better strategies to develop new therapeutics. And one of those new techniques is the development of organ chips. And specifically, we study the gut, so gut on a chip, and using that as a preclinical model outside of the animal arena, but to test various therapies. And how does this particular immunotherapeutic, for example, which we're very interested in our lab, how can we then test those therapies without actually testing them on a human first, but testing them on human tissue? So I'll take you through that a little bit. To start, first, I'll talk about our experimental NEC-like intestinal injury in mice. Just as Hala had mentioned, one of the models that we do is really, we wanted to look at the different risk factors that are involved in disease, and I'll talk a little bit about that in the next slide. But what we do in the lab, just briefly, is we take neonatal mice, we separate them from their moms so they're no longer breastfed, which we know is important to prevent NEC in humans, and they're subjected to a diet that consists of a special formula that's also inoculated with enteric bacteria that came from an infant with NEC totalis, so the most severe form of NEC, and then also other bacterial products, including lipopolysaccharide, or LPS, which David Hackham and Mickey Kaplan's lab have all shown that have been important in the pathogenesis of this disease. And then we also subject them to twice daily hypoxia, because, as we all know, for people that have been in the NICU, either working or as patient families, that we know a lot of our preterm babies do have a lot of episodes of recurrent hypoxia. So really what we're trying to do in the lab is mimic that clinical situation as best as possible. We place them in an incubator since they're separated from their mothers, and then hand feed them, as you can see in this picture to the right here, with special, what we call PICC line, so a special catheter where we drip the feedings in every 3 hours. And at the end of 3 days, evaluate their intestine for intestinal injury and look for different markers of disease. And so when thinking about different models and the biomarkers that come from intestinal injury, what we really want to focus on when we're developing a model is the different risk factors that we know are important in NEC pathogenesis. So we know that our premature babies are certainly the most at risk, but that doesn't mean that our term babies also are susceptible to NEC. But there are several instances in gut immaturity or certainly hypoxia, as I mentioned, that can predispose the intestine to injury. Certainly gestational age plays a significant role. Did a baby or their moms get antibiotics can also predispose differences in the microbiome. There's a lot of good work going on about that in the field. And then also the absence of breast milk, we know is critical for our babies to prevent NEC, but not all of our breastfed babies can certainly, they still get NEC. And so how can we really tease that out? And so just looking at different biomarkers of intestinal injury, specifically, as Hala mentioned, there's a lot of work going on looking at pathogenic bacteria and the microbiome specifically that can predispose an infant to NEC. And what does that do downstream, looking at different bacterial translocation or getting across that gut barrier and really stimulating this inflammatory response that we know is critical in NEC. And so what we can do is really look at different human samples, and then how can we inform those different phenotypes back into our models. And we all know that biomarkers for NEC are desperately needed at the bedside, so we need to do this obviously as soon as possible. And so when we're thinking about biomarkers, where to start then? And so as I mentioned, in 2017, we developed a NEC biorepository with a lot of different samples from a lot of babies. And we're very grateful to all the centers that have come on board with this, and the ones that we're onboarding. We deeply appreciate you as well. But what a biorepository does is that it collects, processes, stores, distributes specimens really to support investigation. And it's easy for us to do our own studies at different centers, but really the importance of this work is really to build capacity in order to study all of our babies that get NEC. We know there's all different phenotypes at all different gestational ages. And so really what we need to do is have a bunch of centers involved. And so that way we can really deeply characterize this disease, with really the goal to facilitate all of our basic science and translational observational studies to learn more about what is actually happening to our babies at the bedside. And I'll just take a moment to talk about intestinal phenotypes that we've been deeply characterizing in our lab, specifically with samples that we've obtained from the operating room, and some studies that we published a couple years ago now, specifically in this slide looking at DNA methylation. And so what we tried to do actually is just take an unbiased, deep multi-omics approach to the samples that we already have in the laboratory. And I won't go into the details of the science here, but what you can see is in these NEC samples here, both in the ileum and the colon, you can see this dark blue here, which is representative of DNA hypermethylation. We have ongoing studies looking at different biological fluids to look to see what the DNA methylation signature is in those, including blood, urine, stool, and certainly intestine of all different types, whether they're resected for NEC or other indications, to see the differences, what's happening prior to disease onset, but also as the disease is actually happening. And then taking a deeper approach, looking not just globally across the genome, but looking at the specific cell types. Our lab and others have done single cell RNA sequencing with samples in human NEC. And these studies are from our lab in collaboration with Jeremy Simon, who's a geneticist at UNC, looking at what's happening actually in each cell at the cellular level during NEC, or in the setting of inflammation or without. And so every piece of intestine that we are lucky enough to be able to obtain in the lab and analyze closer what else can we do with it? And so something that we've done in our lab is develop this neck on a chip model, and I'll walk you through that here. As Hala mentioned, in her lab and others looking at enteroid generation or these intestinal organoids, basically you get a piece of intestine and you can isolate the crypts or the base of the intestine here. You can grow up with various growth factors, etc., these neonatal enteroids. And we have these microfluidic chips that are basically these little tiny environments where you can put cells onto the chip. And as I mentioned before, test various microbiomes or test various therapeutics or nutraceuticals, different feedings, etc., on the chip itself. And how that breaks down, the chip is 2 chambers. There's a top chamber of epithelial cells, there's a bottom chamber of endothelial cells, and the crosstalk between these cells are really important. We also have some work in the lab layering on immune cells as well. That is not ready for prime time yet, but we're certainly working on that. We can add, because of this platform and the way it functions, we can add different microbiomes. So we add this enteric bacterial slurry from an infant with necrotizing, and then evaluate different readouts, including pro-inflammatory cytokines, epithelial proliferation, as Hala mentioned in her talk, and certainly gut barrier integrity as well. And what this looks like under a microscope, this is some work that was done by now a medical student, Wyatt Lonick, in the lab. So Wyatt grew up these premature infant enteroids, seeded them onto the chip, and this is just a picture of that here. And the chip actually undergoes continuous flow of media and stretch to mimic the human intestinal environment. And so you can see there's a monolayer of neonatal epithelial cells here. With that continuous flow and stretch, there's also neonatal epithelium that develops by around day 6. And then by day 8, we have these 3D villus-like axes here. And at day 8, then what we do is we add on our microbiome challenge and then look at different effects on the chip. And one thing that I mentioned is really important is really when we have a model, then comparing that back to the human situation. And so this is a busy slide just looking at different epithelial cell types that are present in human neck and also neck on a chip, and just showing the comparison that in human intestine that had neck, we see decreased goblet cells, PNS cells, enteroendocrine cells, and certainly decreased proliferation or Ki67, as Hala mentioned. And then in our control chips, we see these nice cell types. And then in our neck on a chip model, you see that those are decreased. And again, taking an unbiased approach, we looked here at RNA sequencing of the chips at different time points compared to controls. And so you can see that there is over 5,000 genes that are differentially expressed in our neck and a chip model, several of which overlap with those that are seen in human neck. So there's a lot more work to be done to dissect this as well. But just wanted to show you the different things that we're working on in the lab. And in conclusion, this is obviously, as we're all doing, hard work, and really, just want to stress the importance that neck models are always evolving to a more bedside to bench approach. And together, all of us can do a great job at that. And then also defining the human neck phenotype remains a significant research priority, because we want to make sure that we have the best models possible in order to be able to test various therapeutics or preventative strategies for our babies. And certainly it takes a village to build a world without neck. This is my old lab at Washington University in St. Louis, who did a lot of the work that was presented in this slide. And then my new amazing group at UNC Chapel Hill that is currently working hard in the lab to fight this disease. So thank you so much for all of our collaborators, funding sources, and the opportunity to present today. Thank you.
Thank you, Misty. Wonderful. And I love all the photos of your lab team wearing the Neck Society gear. Thanks for that. All right, Steve, you're up. Thank you.
All right. Thanks, Jen. I will work on bringing this home. pull up these slides quick. Okay, perfect. So I want to thank Jen, the NEC Society, for letting me talk a little bit about what we've been doing in the lab for the last almost 2 decades now. And as I thought about kind of how to do this and talk a little bit about what we do and why we do, I decided to kind of go for an overview approach. And so we're going to talk a little bit about our overarching themes in the lab and different experiments that we've done and what it has impacted and how it relates to the human disease. So again, I kind of wanted to frame this as why I do NEC research and how did I get where I am. And so it will model kind of what Misty and Hala just went through. Similar to both of those investigators, you know, we all have stories. This is a patient that I took care of now 10 years ago, who was born at 30 weeks and was doing great, and then developed really severe necrotizing enterocolitis. She lost her colon and a large chunk of her small intestine, but survived and is thrived, and now is a very spunky 10-year-old. And this is really, you know, these sorts of kids are, I think, what drive us as physician scientists to try and find the answers and figure out why this devastating disease still happens. So I wanted to take a moment and just give a nod to the people that came before us. You know, we do a lot of research, but we stand on the shoulders of giants to reach higher. And so, you know, the very first NEC model that is now kind of accepted was developed by Dr. Barlow in the 1970s. Keep in mind that NEC was really only recognized as a disease process in the mid to late 60s. And what Dr. Barlow did is she figured out that she could do this in newborn rats, and she would either drop or feed them a simulated formula that she had developed, or she would compare it to animals that were fed by their moms. She would expose them to Klebsiella, which is a common bacteria that's seen in NEC. And then she would make them hypoxic by sealing them in, she actually used a plastic bag until the animals were limp. Later, this was then modified by adding hypothermia to the animals. And she would do this over several days, and these animals would develop symptoms that looked like necrotizing enterocolitis. Now, this was modified by Dr. Mickey Kaplan in the mid-90s to move this from rats to mice. And the reason that Mickey did this is because he wanted to be able to make use of the genetic tools that are available in mice that aren't in rats. And this has now been then modified further by Dr. Hackham and is used in multiple labs and has been modified further, as you heard, with what Misty's doing in her lab. So one of the things that always interested me, and a lot of my research has just been driven out of asking questions. And one of the questions was, we know that NEC is developmental dependent. The younger you are when you're born, the more likely you will develop NEC, but it doesn't happen right away. So the kids that are born at 22, 23, 24, 25 weeks of age or of gestation don't develop NEC in the first couple of days. It usually takes them a couple of weeks. And so we know that there's this developmental component that goes with necrotizing enterocolitis. And so we asked a simple question in the lab, or what seemed like a simple question at the time, which was, if you look at a mouse, can you find an age in a mouse that's similar to that age in humans where they develop necrotizing enterocolitis? And this was a simple question that turned into, I think, a 3-year big project. And I have to give a nod to Misty because she helped a lot with this. But essentially what we found was that it does make a difference. And at least for the epithelium, mice that are somewhere around 2 weeks of age are probably similar to humans that are around 24 to 28 weeks of age. And mice that are earlier, at least from an epithelial standpoint, are more akin to pre-viable humans, and mice that are later around the 4-week age are similar to humans born at term. And so this was a question that we asked, and was helpful because it allowed us to pick which age of mice we want to look at, depending on the question that you're trying to ask. So Hala already mentioned this. One of the other things that we asked early on was, is there another way that you can develop NEC? And this came out of a question that I got really when I was presenting a different piece of science at a large national meeting. And I was asked about panacell biology by one of the audience members. And for all the trainees, I always say this is a really important lesson, because that simple question and taking it further has led to really about 15 years of research and the development of this new model of necrotizing enterocolitis. And so what we did is we realized that if you disrupt panacels, and panacels are important cells at the base of the intestinal crypt that really serve as the watchdog or the gatekeeper of the intestine. Their function is to prevent bacteria from getting into and around the epithelial surface, or the surface lining of the gut, and also to provide health and healthy growth factors to the stem cells that surround them. So if you disrupt these panacels, and then you follow that with some sort of dysbiosis, or changing the microbiome to a more inflammatory state, you'll develop something that looks like necrotizing enterocolitis. And you can see in the pictures here, we're injecting a mouse with a chemical that we use to disrupt those panacels. We're feeding them. This is how you feed 2-week-old mice, with a little needle. It's a blunt tip, so they're not actually hurt, but it is a little tricky to learn how to do. And you can see the intestines here are really typical of that necrotizing enterocolitis. And you can see some bubbles of pneumatosis through here, and the patchy appearance of NEC that we associate with the disease. But what is more important with this model is that it does carry that age dependent dynamic that NEC has. So if you do this in mice that are 5-day-old mice, you can't develop the disease. And if you do it in mice that are 28 days old, you can't develop the disease. But if you do it in mice that are 2 weeks old, you do develop the disease. And so it's really been a nice model for us to work with, because it carries some of the really important characteristics of necrotizing enterocolitis, not only the injury, but the timing as well. And a lot of people have asked throughout the years, why does this matter? This matters, we've found, because panacels do seem to play a role in human NEC. When you look at babies that have developed NEC, and you look at their intestinal tissue after the disease, they have decreased numbers of panacels, similar to our model. What's further, Venki Sampath's lab has done some nice job showing that humans that have genetic mutations in their panacell biology pathways are also associated with increased NEC development. So we know that these panacels are important to disease process in babies. And so this model has a nice link to the kids that are developing disease. The other big question that we had wanted to ask, and are currently asking, is this. We know that moms that have infections right before birth, or chorioamnionitis, are more likely to develop preterm. And there's also a known association with those kids developing necrotizing enterocolitis later. And the more severe the mom's inflammation and infection, the more likely you are to develop NEC. But nobody has really been able to understand why this happens. If the inflammation was causing damage to the gut immediately, you'd think the disease would happen soon, but it doesn't. It happens weeks after birth again. So this was a really interesting question, and it was actually asked by one of the trainees in the lab that got us thinking about how we could follow this up, and how we could try and answer this question. And so what we did was we took models of this chorioamnionitis, or maternal inflammation, And we modified it for our lab. And so we inject mice with lipopolysaccharide, which is a piece of the cell wall of gram-negative bacteria. And that induces inflammation not in the mom originally. And it's a really significant inflammation. But more importantly, it continues to cause inflammation to the neonatal offspring. So when they're pups, so we inject the mom about 4 days before she gives birth. We just let her deliver normally. And the offspring that are born also have significantly increased inflammation compared to animals that aren't exposed to this. And we have coined this maternal inflammation model, FEMI, or fetal exposure to maternal inflammation. So why do we do this? Well, the next question was, what happens to the intestines of those offspring? And not doing anything to them, what we found is that the animals that were exposed to this FEMI were having much more increased inflammation in their intestines and injury in their intestines. And not only were they injured, but the injury lasted and persisted. So what you can see off on the right here is a picture of intestinal injury. And we scored this as either a 0, a 1, or a 2. If you're a 0, you're relatively normal. If you're a 1, you have kind of mild-ish injury. And if you have a 2, you have more severe injury. And you can see that the animals in the red, which were the ones exposed to LPS, were all higher than the animals that were normal. And again, this is without having any secondary exposure. This is just animals running around and acting normally. So we then took this a step further and said, well, what happens if you give them injury? And this is research that was done by Aaron Fricke and Tim Elgin in the lab, as well as Brian Juber and Sarah Watson. And what we've found over time is that there's a disruption of those PANA cells. So going back again to the theme of PANA cells being important, when you are exposed to this maternal inflammation, you can see here the animals that are not exposed are in gray and the animals that are exposed are in red. And as you go through time, P stands for postnatal. So these are 14-day-old mice out to mice at 56 days. Over time, there's an increasing drop in PANA cells compared to normal. And what we think that's doing is that's setting up the likely chance that you're going to have injury. So as Hala mentioned, our NEC model is made up of 2 hits. So you can't have just one hit, you have to have both of them together. One is a PANA cell disruption and the other is a dysbiosis and a inflammatory surge. And so what we think is happening is that these animals that are exposed to the FEMI exposure, like simulated chorioamnionitis, are having decreased numbers of PANA cells, which then allows them to develop NEC more easily. And you can see off on the side here, what we found is that if we expose those FEMI-exposed animals to just one hit, which is the dysbiosis, they will go on to have injury. They don't need both hits anymore. And so it's things like this that allow us to take and answer questions that are really pertinent to what we see every day in the NICU, but don't have answers to. And by utilizing these animal models, we can really start to answer those questions and move our knowledge base forward. As Misty and Hala said, it really takes a village to get these things done, and I can't do this by myself. And so you can see pictures here of lots of trainees that came through the lab, as well as our collaborators and our funding sources, which are all super important to be able to really move this knowledge forward. And with that, I will end this, and we can move to the question part.
Awesome. Thank you so much, Steve, for your excellent work and presentation. Really wonderful. So yeah, if everyone can turn their cameras on, that would be great. And we will get started with, first, we're going to just have a panel discussion and let our group kind of react to each other's presentations. So if you have any questions that you want to start with, that would be great. And then once our kind of team right here is finished, we'll transition over to our Q&A box. We have one question that's been submitted that we'll answer live. And if anyone in the audience, if you have questions that you would like us to address, please put it in the Q&A box in the Q&A tool, and we'll get to it in just a moment. So I guess the first question that I'd like to pose to Hala, Steve, and Misty, whoever wants to take it first, is just how do you see patient families being able to help accelerate the basic kind of modeling neck research that you're doing?
So I'll go ahead, Misty.
No, you
I'll
go.
make you go first. I mean, so I have been fortunate to be with the NEC Society for many, many years. And what I can say is what is really cool about having parents involved and how it accelerates things is we hear what's important to them. And it helps color. Sometimes, neck is a really complicated disease and there's lots of things to look at. And sometimes you can get really focused going down a rabbit hole. It's nice to have parent input because it helps bring you back to what's important at the end of the day as the kids. And so it helps drive things, as well as giving us some insight as to other things that we may not be thinking about and can bring up questions that we can answer down the line.
Thanks, Steve. I was just going to add, I think, from a NEC Society standpoint, at least over the last, several years now, there has been increased collaboration in the field, which I think has been incredibly important, but also, as Steve mentioned, making sure that we know what patient families are really looking for. We know like diagnostics and therapeutics, of course, are really important, but I think also, how like with the feedback we've gotten on consent forms and what is really important for our families when we're thinking about the biorepository and utilizing those samples for biomarker development, et cetera. I think that's been really instrumental, at least from a NEC biorepository standpoint. I think it's just really awesome, all the amazing work that you're doing.
I'm going to add to what Misty and Stephen mentioned with really something that was very impactful for me maybe 4 years ago, it's been maybe longer, is when the parents actually said how they want us to talk to them. Like how not just as a researcher, but as a neonatologist, is not to sugarcoat things, to just talk about how dangerous neck is and what would be something that we should be looking for. I'm also these stories kind of brings it back home to we need to do more. We need to be more passionate. We all as researchers go up and down in how funding and all the stuff that happens and why do we do what we're doing, and having the NEC Society and the parents' feedback and all the stories about whether it's the death of a patient with NEC or survivors with outcomes that we didn't even think about. I think this is what probably is going to make us move forward to not just look at death or neurodevelopmental outcome, but actually how it impacts the families. So the more we hear, the more we're going to be excited about doing what we're doing. And, these frustrations with, for example, not getting funding is just going to go away. We'll just continue to charge, hopefully, and make it better for everyone.
Those are 3 incredible answers, and it does not surprise me one bit that these folks are part of your team, Jen. You've built such an inclusive team. I want to highlight that I 100% plus agree with all of those answers and how patients and communities can really help us all focus. I think what's most important is that the organization then serves as the collector and the distributor of that information, so that information doesn't just sit in a vacuum in Steve's lab or Misty's lab or Hala's lab, right? And so being an active advocate, patient, or parent is so crucial, but making sure that you share that information with the foundation so that it can all be collected and really exponentially become more powerful because you're hearing so many voices at once.
I love that, Andrea. Thank you so much. And thank you to Steve, Hala, and Misty for believing in the patient family voice and perspective, and just like recognizing the value that families can bring to research. I think it's really amazing and transformative. Do you have any questions that you'd like? I mean, I can keep asking you questions, but I didn't know if you guys want to pose any questions to each other before I jump in with another one.
No, go ahead.
All right. I guess the next question I have for anyone who wants to answer is like, where do you hope next science will be in the next 5 years, 10 years? Like, obviously our long-term goal is to build a world without this devastating disease, right? That's our vision. But how, like, how are we going to get there incrementally and, step by step? Like, where do you think the science will be in the next 5 to 10 years?
I'll take that. Mostly because I'm the big dreamer of the group. I would say, at least in 5 years, we have now have 10 centers, and we're onboarding more into the NEC biorepository. So with all of those patients with NEC and control across, all over the US, I really feel like we have this enormous opportunity to really figure out some biomarkers. I know there was a question in the chat that I was trying to answer, but just the question was specifically asking about progress in biomarker discovery for NEC. And I do think we've made a little bit of progress, but not as much as we really should. And so we need to be doing transformative science in that regard and really deeply characterizing all the samples that we have in everyone's freezer to really come up with some answers about what's exactly happening to our babies at the bedside and how can we detect it sooner and intervene earlier.
Yeah, I will. I'll piggyback off of Misty. In watching where NEC has gone since Mickey was really doing his first stuff, we've gone from being a small group of people trying to find answers to having this biorepository where we have cross communication and we can start looking in a large way at the things that we're finding in the lab. And so I agree. And I hope that kind of continues so we can move forward. I think Misty's typing about the biomarker piece. We don't have one. And it's really hard because we can't tell who's going to develop NEC and not. I know there's a question about computer simulations and virtual models. All that stuff depends on us being able to say what NEC is. And in order to do that, we have to have these biomarkers. And so we're still in that process. And so I would hope in the next 5 to 10 years, we can go beyond just trying to determine what NEC is and having a better definition so that we can then start using the computer modeling, start using AI to try and narrow things down. And I think that is hopefully where things are going to be moving.
Yeah, I love that. Thank you. I guess one question I have that I want to build off of what Hala said is when the baby that she was caring for passed away, the kind of response was like, well, NEC happens. And I will say, like, that is what I was told when Micah developed NEC. Like, he was doing really well and was progressing and was, I thought, would be coming home in a couple of weeks, and then developed very severe NEC and had multiple bowel resections. And the kind of response from the team was like, yeah, that just happens sometimes. Like, NEC just happens. And I have always found that to be a really unacceptable answer, that it's like kind of inevitable. And so I'm curious, like, Hala and to the rest of the team, like, do you feel like NEC is kind of inevitable? Like, I mean, there are ways that we can and have to do more, right? Like, I'm sure that's why you're here. So I just want to see if you can respond to that kind of attitude and if you feel like that is changing and how we might be able to help that shift.
I'm going to start because I completely agree with you. I actually thought that was not acceptable. So that's why I kept on going back. I'm like, I'm in the United States right now. Like, you can't tell me that's inevitable. Like, this is going to be happening anyway, and that's NEC. But to be honest with you, we've all had these preterm infants that were doing so well. And yes, we have a lot of risk factors that predisposes them to developing NEC. But for example, we've done exclusive human milk feeding right now. We've done fortification with human milk. We try to reduce antibiotic exposure. And then sometimes we have those babies who develop necrotizing enterocolitis. And I believe that some of what Misty is doing right now, which is identifying, and actually what Dr. Sampath have done, is identifying the genetic maybe predisposition toward why they develop NEC, but looking into maybe some other methods of the microbiome. Is there anything we can do to shape the microbiome for infants who are at higher risk? Going back to what Stephen mentioned, which is the big data and the bioinformatics, to actually pinpoint the one that are really at higher risk, maybe trying to help that. But yeah, I agree with you. It's an unacceptable answer. And I think we're all here to try to figure out what we can do in reality, not just in the models, to be able to stop that answer, which sometimes seems to be pretty harsh and cruel, is always harsh and cruel.
I agree. I think just thinking that it's inevitable is really unacceptable, and it's a closed mindset. And we have to really move past that and really drive change in every NICU, And I know there was a question in the chat about strategies people could implement in the NICU for registered dietitians. I think, human milk is one of those things. It's one of the only things that we have to really, encourage our families, to do if possible, or provide donor milk. So I think just ensuring that, there's other things, obviously limiting our antibiotic use and things that we do in the NICU all the time. I think just paying attention to those things, following a feeding protocol, things that other investigators have shown have been really important in the prevention of NEC. I think those are all things that we could be doing in our own centers.
Yeah, and I want to stress too, it is why research is so vital, because at the end of the day, we're dealing with a immature intestine. And I often describe NEC as like you're getting on a plane and you're flying it, but I've taken, 2 out of every 3 screws out of the plane. It's a formed intestine that works, but it's not completely formed. And that's really the end result of why NEC happens. And unless we have more research to really understand why that immature intestine is different and why that immature intestine is susceptible to NEC, then we're no better off. NEC did not occur until we started saving preterm kids. And so it's inevitable in that it is a function of being premature. That doesn't mean that we can't impact it and help it and prevent it from happening. But we've got to understand the landscape that NEC occurs in order to be able to do that.
That makes sense. I would love for us to answer some of the questions that have come into the Q&A live, and just kind of, I know some of you have been typing in answers, which is definitely appreciated, but wanted to see if, maybe the first one that I said we would answer live is just on cardiac NEC and babies who have congenital heart defects and then end up getting necrotizing enterocolitis, and just any thoughts or comments that you want to make around babies with heart defects in NEC.
So I'm going to start with this, and then everybody else can chime in. So one of the tricky things with NEC, and one of the reasons why we don't have a good definition, is that it's likely multiple pathways that end up in the same phenotype or the same disease state at the end. And cardiac NEC is kind of the prototypical backing for this. We know through studies that the cardiac NEC is a little bit different than preemie NEC in that it's more blood flow driven versus inflammation driven. And so it's really important. There's actually not a lot of good basic science on this. Most of what we have is through what we know clinically in kids that this is developing in. It's an area that is ripe for research. It's difficult, but I think that it's, again, one of these areas that we need, as we continue to move the research forward, we need more people looking at this and trying to find out more about it.
And it's something that the NEC Society is really committed to. Thankfully, we've had patient families that have come forward to advocate that we really shift our focus towards this area of term babies and heart babies who have NEC. And we've recently are continue to just amplify those efforts and encourage people to do the research and to really better understand how and why this happens and how we can prevent NEC in babies with heart defects. So thanks for that question. Missy, I know you mentioned just about the disease of neck and like using biomarkers to predict and prevent, but didn't know if you wanted to expand on that answer at all, just around the progress that's been used with biomarkers.
Sure. I think I think the short answer is there hasn't been a lot of progress. I think a lot of the studies, including some that, you know, have come out of my lab, have been smaller pilot studies, right? And so we're collecting the samples and we're trying to analyze them as quickly as possible and get something out there that could potentially help the field. But I do think there is something to be said about gathering enough patients that can give us some answers. So I think that's why it's really important that we all work together on this, because you may have 6 to 8 to 10 babies at your center that have, let's say, surgical neck or the most severe form of neck. But a small cohort like that isn't going to necessarily give you the answers that are necessary to tease out what's happening at the different gestational ages. Is this something that's developmentally regulated, for example, because we see in preterm neck, we see, you know, this developmental window between like 28 to 33 weeks corrected age, where there's obviously something happening there, but how can we deeply pinpoint that? That all those things are really important, especially cardiac neck, because all we know is that really we think, because there are no good models of congenital heart disease neck models, all we really know is that we think it's related to decreased gut perfusion, which we know, at least in our mouse model, predisposes the animals to neck, but we don't really have the greatest phenotype, I would say, on those babies. Why that is because they're rare, but we know that they happen. And so certainly, you know, we talk about neck being a rare disease that we all see all the time, but then when you subcategorize that, as Steve mentioned, into different categories, and what is true neck and what is feeding intolerance and what is milk protein allergy and what is true cardiac neck, I think, you know, in order for us to get a biomarker, we have to really make sure that we're phenotyping these babies that are getting neck at the bedside correctly, because our diagnostic tools are really not that great. And we all know that we see abdominal x-rays or abdominal ultrasounds. It's like could be pneumatosis, could be, you know, and could be stool, and that really is impactful for the baby in their hospital course. So I would just advocate that we all continue to participate in research and help each other really in order to advance this disease.
Yeah, thank you. Are there any others that would like to expand on any of the questions and answers in the Q&A box that you want to talk about live? Thank you for typing in answers to most of them. I do have a, yeah, go for it.
No, I was just going to say, I had a general comment, there was a question in the chat about neck on a chip models, but really all these models that we use, all the different animal models, the enteroid models, the neck on a chip models, all of those are just that, models. And so it's not something that we would necessarily you know, be doing at the bedside. Some of these models are actually really expensive. So the piglet models, for example, that David Hackham's lab and Gail Besner's lab uses, those model, and the neck on a chip model, for example, these are really expensive models. And so, so it does take a lab with considerable resources, which isn't that great for advancing the science. And so I think as the costs come down for different items like sequencing, you know, these microfluidic chip devices that we use, as those costs tend to come down, then we can see the science really advance. That's something that we've seen with all the multi-omics approaches that start out really expensive and then get a little bit cheaper as they become more mainstream. I think all of us that are doing research, just continuing to push that agenda and envelope is really, really critical. I think recognizing that there are models and they're preclinical, and once you translate, you know, drug, for example, to the bedside, you know, can really impact our babies in a different way. So I think thinking outside of the box and strategizing in that regard for all of us that have labs, I think is really important.
Yeah, excellent. I see that there's a couple of hands raised, by the way, so feel free if you're an audience member, feel free to type it into the Q&A box or the chat box. But yeah, Andrea, go for it.
I have a question, Jen. I hope you don't mind me posing a question instead of answering a question. One thing I think that the NEC Society does incredibly well is acknowledge the importance of representing everyone. And really reaching a diverse population of patients. So I'd love to hear how I just love to hear from the physicians about the importance of this at the bedside and really contributing all stories and all phenotypes to helping us really understand and maybe identify a biomarker.
So I think it's an important question, and I think I think of it in terms of steps. So in order to have all people included, that's the power of what the biorepository is doing, right? Because we can get all swaths of life, all areas of the country, and that's really powerful. But before we can do that, we have to go back and do the preclinical stuff. So all of the various models that the three of us talked about allow us to then go to something like the biorepository and then start to look at this. You have to find out and understand how the processes are working in an animal model before you can then take it to human samples and validate it and see if it holds true. Otherwise, you're essentially looking for a needle in a haystack, and you're taking really precious human tissue and kind of running shotgun stuff, just trying to hopefully find something. And so I think that's where the power of this is. It's just we're still in that intermediary step of trying to figure out how NEC happens to then take it to the biorepository level and then start to really actually tease out these various things. The biorepository has been great at this. Some of the other genetics studies that have been done have been really good at this. But we're still kind of in that in-between phase.
Yeah, I would definitely add on that. I think one of the most frustrating things at the bedside is really being unable to provide much hope, as I kind of mentioned. Although I just say we're really trying, we're working on it at my center, but that doesn't mean like, another family at another center without a NEC researcher doesn't have that type of hope or even opportunity to donate their child's, samples for this work. So I do think something like that is, it's really important. And that's why we're doing the biorepository and trying to offer some hopes for all the centers.
And I agree with Misty. It's really frustrating. I mean, I'm sure Misty and Stephen, you do the same thing. I'm, considered the local expert for NEC over here. And then usually when a patient develops necrotizing enterocolitis, it's the most frustrating experience to have, which is trying to explain to the family that this is not something, for example, that you hear, is it something that happened from pregnancy? Is it something that I was eating and maybe my breast milk? Like you just have to sit down with the families and ask what questions they have that could be very uncomfortable for them sometimes. And unfortunately, which I go back to what Jen mentioned, is sometimes we say, unfortunately, your baby is premature. And if we can prevent prematurity, we can prevent all premature diseases, including preterm NEC, though it won't prevent cardiac NEC, for example. But it's very frustrating. And I agree with Misty, is what I usually try to do, and that's what I sometimes reach to Jen. Actually, I do reach to her. It's like there's a family, they're having a hard time, processing what's happening, and we try to provide as much as we can, but having that other family perspective to let them know that there, unfortunately, someone else has been there, and unfortunately, I wish we can prevent it at this moment, but they're not alone in there. They can have questions, they can ask more things, they can feel a little bit more empowered about asking for the next steps, which I think is very important, is having this power of what do I need to know to ask? which is very helpful to them. So I definitely want to thank the NEC Society for having that aspect that I think we cannot provide at this moment, is the family perspective and the ability to talk to them freely without feeling the questions they're asking are maybe too simple, if that makes sense.
Yeah, absolutely. Hala, thank you. And we're really honored and proud to be able to provide resources to families who have just been diagnosed or who have tragically just lost their baby to this disease. And we actually have box materials that we send to NICUs to provide to families that are just going through this. So yeah, it's really an honor to be able to do that, because it's an incredibly isolating experience to go through this really devastating disease. The time has flown by, and I have a few closing slides. Any closing thoughts from you all before I share my screen with our closing slides?
Just thank you to everyone for being here and listening and supporting the NEC Society. And thank you to CZI for also caring about our work that we do. And we're really thankful to drive this work forward.
Yeah, awesome. I'm going to share my screen. So just want to highlight a few ways that you all can stay engaged with us. As I mentioned, we have a lot of resources on our website. We've done over 15 webinars now. The recordings are available. This recording will be available soon. We have lots of resources for families who are just being affected by this devastating disease. We have a webinar coming up in October, which I'll share the registration link in just a moment. My colleague will in the chat box. We encourage you to subscribe to our blog, follow us on social media. We actually have an in-person meeting planned for next year, which is very exciting. We've been virtual during the entire pandemic. So we actually haven't been in person with our community since 2019. So we're very excited that we're going to bring everyone together next year. And you can also support our work and help us build a world without NEC at necsociety.kindful.com. So here is our announcement for our next webinar, which will be focused on surgical NEC, so babies that are needing valve resections. It'll take place on Friday, October 14th, and will feature a lot of the surgeons that are really critical to the work that we're doing at the NEC Society and helping us build a world without this devastating disease and improving outcomes for babies that are diagnosed with this NEC disease. As you saw in many of Misty's photos, we have excellent t-shirts and gear in our NEC Society shop. It's a great way to support our work and help spread awareness. So you can visit our shop at necsociety.org/shop. I encourage you to check that out. We're super excited to be launching a nurse ambassador program, which we'll be launching in the next probably couple weeks. And it's a way for us to encourage and engage more nurses at the bedside to engage with the NEC Society and help us spread awareness and prevent this really devastating disease and empower families with resources and information. So stay tuned for that announcement. Many of us will be running the California International Marathon on December 4th. And if you can't be with us in person in Sacramento, we encourage you to register and to run and walk in your own community on December 4th. So that link should be showing up in your chat box, and we hope you'll join us for that. And finally, this is our first announcement that we have our next symposium taking place in San Diego, August 28th through the 30th of 2023. So please mark your calendars, and we really hope that you will join us in San Diego for this really exciting opportunity to bring our community together for the very first time in too many years. And again, just a friendly reminder that our work is going to be focusing on our research priorities. A lot of the work that will take place at the next symposium will focus on accelerating the research priorities that we've just recently identified. So we look forward to having you help us accelerate this work. And again, thank you to Charlie's family for allowing us today to keep this webinar to her, and to all the patient families that continue to share their stories and their voices with us, and of course to the clinicians and the researchers who have just dedicated their entire careers to advancing the research and the work here that we are doing today. So thank you all so much, and we look forward to seeing you again soon. Take care, everybody.